Hello, this is Jack Clements, and today we'll be discussing the mitotic process as a student-prepared poster board. The first part of the mitotic process is the preparation for the mitotic process through interphase. The first phase is prophase, and as you can see here, in prophase, the chromatids condense into chromosomes. The nuclear envelope breaks down, as you can see here. This is the condensation and the nuclear envelope fragmenting right here. It is still in form at this point. It is starting to fragment. These spindle fibers emerge from the centromeres right here. Spindle fibers is kind of the cross webbing you see here. And as I said before, the nucleus disappears. The next phase is prometaphase. In prometaphase, the condensation of the chromatids into chromosomes continues. The kinetochores appear at the centromeres. So that's, as you can see here, these little kind of darker spots in the middle of the chromosomes. That would be the kinetochores. The microtubules connect to the kinetochores, so that would be this kind of connection right here on top of the spindle fiber. And the nuclear envelope is fragmented completely at this point. It is kind of surrounding itself around this part of the chromosomes. The next phase is metaphase. And in metaphase, the mitotic spindle is developed. The centromeres are at opposite poles, here and here. The chromosomes are lined up at the equilateral plate. As you can see here, that is just kind of line running through the middle. The chromosomes are lined up at this plate, and the sister chromatids are attached to the spindle fiber itself. Okay, and the next phase is anaphase. In anaphase, the cohesion protein binds sister chromatids, and they're pulled to opposite poles, and it separates these chromosomes from each other. So the parent cell will form two daughter cells with the same amount, and at this point in the mitotic process, you see that they are pulling apart into opposite sides of the cell. Also at this part, the spindle fibers kind of move back, so it elongates the cell and prepares it for the final stage. And the final stage is a telophase followed by cytokinesis. And in telophase, the chromatin decondenses at this part, so we're now decondensing and we're elongating into chromatin. The nuclear envelopes are starting to reform, and then you begin to see what is here is a cleavage furrow, and this will be the site where the cell actually separates. And then this would be the final product. You'd have two daughter cells with the same number of same amount or number of chromatin as the parent cell. This concludes the presentation. Thank you.